we go. All right. Hi, everyone. Going to give it a minute to let everyone come on in to our cafe. And while we're waiting, I'm just going to get my slides open and see who's coming on in today. Just give me 1 minute here. Got to move things around. Hi, Jack and Simon. So you guys know the drill so well. All right, for those of you who are new to our Teen Science Cafes, I'm Lauren Traster. I'm the 4-H Teen and Leadership Program Coordinator here with Vermont 4-H. I'm going to pop into our chat. If you need our live captioning, just click on that link so that you can have our captioning services today. So for those of you new, we like to introduce ourselves, and I love seeing everyone coming on in today. Mariah and Maddie and Emily and Brooke, welcome everyone. So open up your chat, go on in and make sure you select all panelists and attendees so everybody gets to see who you are and where you're from. We just like to say hi to everyone. I'm going to put the live captioning link in again. And it's so nice. I, again, recognize so many names from previous cafes. So it's always nice to have our old friends as well as our new friends joining us today. So keep introducing yourselves. And I'm going to just remind you all of our protocols when we are here hanging out in Zoom land. Everyone is muted today. But that does not mean we don't want you to share your thoughts and ideas. And there's 2 ways that we do that. We use the chat box, just like you all are using right now. And the chat box is mostly used to respond to the presenter. The presenter will often ask you questions, eliciting feedback from you. You're going to use the chat box. You can talk about the topic with one another, but really make sure that the chat box stays on topic and doesn't end up being a distraction. If there are a lot of off-topic chat going on, I will privately message you and let you know that you need to focus a little bit more, because we want to be courteous and respectful to everyone. And when we have the chat going too much and it's not on topic, it really does become a distraction for a lot of us. But again, we want you to all participate and engage. And so we use the chat box that way, but we also use the Q&A box, and you'll find the Q&A box in your menu. And the Q&A box is where you're going to put your questions for our presenter. The presenter will decide when she has a good break in the presentation to answer your questions. So it might be all at the end, it might be scattered throughout the presentation. But just make sure to be specific with your questions so she understands, really what you're asking, because it might be, 5 minutes after your question was written and it might not connect to a slide. So just think about that. So we are going to get to today's introduction in a second. I wanted to make you all aware of 3 different programs that are new to 4-H. They're in our 4-H Teen Time series. So we have one that's on career exploration that's called Designing Your Future. And we had our first session today, but they're recorded, so you can always catch up. It's really interesting to be thinking about career pathways and your skills and interests, and then how to navigate and network and explore different jobs. So if you're interested in that, you can sign up to join us next week. There's also an opportunity called Shaping the Future. We're offering full scholarships to 8 teens, and you do have to be from Vermont to qualify for this, and in grades 9 through 12. But the National 4-H Conference is a virtual conference this year, and it's a really interesting opportunity because they have roundtable discussions around all different kinds of issues, and you get assigned one of the issues with kids from all over the country, and you work on that issue, and then you present what you came up with and what your solutions might be to people who actually work in those government agencies. So if you're someone who's thinking about, issues and causes and wanting to influence people who work in the government, this might be an opportunity for you. It is an application, and you do need to apply by next Wednesday. So you can learn more on the link that is on this slide. And then we just posted a program called Critical Conversations. And this is going to be a guided dialogue to help teens really tackle some of these big issues we're all talking about these days, like racism, implicit bias, equity, discrimination. And through this guided dialogue, you're going to learn how to facilitate these difficult conversations. So that might be something some of you might be interested in. So I wanted to share those opportunities with you. But now I want to get to today's presentation. I see, so I'll put the link in the chat for everyone if you want to go explore those opportunities. Once I'm done talking, I'll get that into the chat for you all. So today's presentation is, sorry, I gotta call this up. I'm having technical difficulties. Hang on. All right, here we go. So we are going to be learning about ruminant nutrition, the symbiotic relationship between the cow and her, I can't see that word, and her microbes. Thank you. And our presenter today is Lauren Baker. Lauren is a graduate research assistant who's getting her master's in animal science from the University of Vermont. Lauren received her undergraduate degree in animal science from Tarleton State University in Texas, where she was an undergraduate researcher evaluating the fiber content of disposable coffee cups as an alternative feed additive in the rations of cattle. And her current research seeks to evaluate the effects of dietary yeast supplementation on rumen microbiota and their functions. So let's all welcome Lauren. And I don't know about you, but I love cows, so I am really excited to learn more about them. So welcome, Lauren. We're glad that you could join us today.
Hi, Lauren. Thank you. I appreciate you for the introduction. Hi, everybody. Give me just a moment and I will share my slides with you. Okay. All right. So as Lauren introduced, my name is Lauren Baker. I'm a master's candidate from the University of Vermont, and I'm currently getting my degree in animal science, and I study the rumen microbiota of cows. And so today I want to talk to you about ruminant nutrition and what the symbiotic relationship between the cow and her microbes. So before we really get started and dive into this, I kind of wanted to just give you a brief outline and talk about what we're going to discuss today in this presentation. So I'm going to start off by giving you a little bit of my background and how I got to where I'm at, and then touch on ruminant animals as a whole, and then kind of dive into what rumen anatomy and ecology is, the importance of the cow and her microbes, and then understanding rumen microbes, what they can do and what we can do to make them perform better. And so that will kind of touch into my research and what I'm doing currently. So a little bit about my background. In high school, I was very involved in FFA, which is Future Farmers of America. I'm originally from DFW in Texas, Dallas-Fort Worth area in Texas. And so I spent most of my high school years doing FFA and got really involved in agriculture. And so ultimately, I wound up going to Tarleton State University, which is a sister school of Texas A&M. So that's about right here in Central Texas. And there, I really didn't know what aspect and what I wanted to do as far as agriculture was related. I just knew that I really enjoyed being around animals and was very interested in how I could make a career out of that. And so ultimately, my sophomore year of undergrad, in my undergraduate career, I went and volunteered at our Southwest Regional Dairy Facility. So here you can see at the bottom picture, that's a picture of our facility that we have in our town. And this facility essentially is a small model built for the community to be able to understand how dairy production works. And research is also conducted here by graduate students from the university. So my sophomore year, I went and volunteered at this dairy facility where I met up with a graduate student who was conducting her own research project there and really got interested in dairy and dairy cattle specifically. And so from there, I applied for an internship in New Mexico called the United States Dairy Education and Training Consortium. And I was able to go there for 6 weeks and basically explore all of the large dairy facilities all around the Texas, New Mexico area. And speak with dairy farmers and also have courses related to nutrition, animal welfare, animal breeding, and different topics of that nature. And I really started to discover that I really liked animal nutrition and the aspects of how different the ruminant animal is and what we can do to kind of facilitate the agricultural and production side of that through manipulation of nutrition. So that's kind of my background as far as my undergraduate career. And so after I got back from the consortium, I started as a undergraduate research in the nutrition lab. Like Lauren said in my bio, I evaluated coffee cups, reusable coffee cups as a fiber source in dairy cattle rations. And that from there, I decided that I wanted to pursue graduate school in order to further my knowledge of ruminant nutrition. So that led me, yes.
Can I just interrupt one moment? You've used the word ruminant many times, and there is a question about what does ruminant mean?
Yes.
Could you just cover that quickly?
Yeah, okay, I'm sorry about that. A ruminant animal is a herbivorous animal, an animal that eats forages. And essentially that animal who consumes these forages has this compartment in their stomach in which they're able to use rumen microbes to ferment these forages that they consume. So that is what a ruminant animal is an animal that contains a rumen. If that helps explain a little better, which I have the definition in later slides. Does that answer your question? Okay, sorry. Sorry, so I went to graduate school at UVM where I'm studying ruminant nutrition, and I'll touch a little bit about what's happening in these photos. But essentially, I am currently finishing up my degree right now in which I've already conducted my research. And then, so I'm writing my thesis currently. So this is the question that I was going to ask all of you is what exactly is a ruminant animal? And since I kind of already went over that, we'll just go to the definition. A ruminant animal is a herbivorous animal that contains a rumen, which is a pregastric chamber able to derive nutrients from plants through microbial fermentation. So I kind of wanted to take a poll from you guys. Aside from cattle, which of the below animals are also considered to be ruminants? We have A, horse, B, sheep, C, pigs, and D, chickens.
And I have launched the poll, so hopefully you see it and you can put your answer in. If for some reason you don't see the poll, then just put the letter in the chat box.
Perfect. I do see the poll. It looks like we have 18 of 25. All right.
All right, it looks like most of you have answered with a good 71% saying sheep.
Yeah, awesome. So that is great. So that whoever answered B for sheep, that's true. Sheep and goats are smaller ruminant animals, and cows, pigs, horses, pigs, and chickens have a different anatomical structure for feed digestion. So those of you who answered sheep, well done. Good job. Okay. So just to touch on a few other ruminant animals that you might have heard of. So like I said, goats and sheep are both smaller ruminant animals, as well as deer, moose, giraffes, and buffalo. And these are just a few. There's a few more as well, but just to kind of give you an idea of these animals, what a ruminant animal is. Okay. Are there any questions so far before I keep going?
We're all set right now.
Okay, perfect. All right. So I wanted to touch to you guys on the comparative anatomy of digestive systems of ruminant and non-ruminant animals. So right now we're looking at a ruminant animal, the cow. And you can see when the cow eats her food, it goes through the esophagus and into this rumen. And this is the pregastric chamber that I was talking about, where all the microbes will live and ferment the feed for her. And then it will go through the omasum and abomasum. And this is the stomach that's much like ours, that contains glandular secretions of acids and stuff. This is so once it goes through this process, then it'll go to the small intestine, and that's where you're going to get the absorption of most of your nutrients from your feed digestion. So if we look at a non-ruminant animal, this is called a monogastric animal. That's what we are as humans, monogastric, mono meaning one, gastric meaning stomach. So you can see here, anatomically, we're very similar to pigs. And here, the mouth, when this pig eats its food, it's going to go into the stomach and break down using those secretions and acid secretions to break down the food. So they don't really have the pre-fermentation process that occurs through these microbes in the stomach like a cow would. Okay, so just to touch, ruminants have 4 compartments, i.e. rooms. And I say rooms because there's a large misconception that cows have four separate stomachs, and they don't. They have 4 different rooms or compartments. So much like your house, you have your house as a whole, and each room has its own function, but you wouldn't consider your living room to be your house as a whole. And so that's the same with this, is that each of these are different rooms, the rumen, the reticulum, the omasum, and the abomasum. And the abomasum, like I said, is the true stomach and the stomach that we have that has acid secretions. So if we're looking at a size, so a cow is a very large species, say, compared to a goat or sheep. And so if we look at this, the rumen is approximately 40 gallons. So that's about 20 times the size of the reticulum and about 10 times the size of the omasum and abomasum. And so 40 gallons, to give you an approximation, is essentially the size of a bathtub. So if you were to fill this bathtub up, that's how much room that this animal has in her rumen for all this feed to be digested. And then you have the small intestine, which absorbs all these nutrients at 15 gallons and 10 gallons for the large intestine, respectively. So just to talk a little bit about rumination, I don't know if you guys have ever actually watched a cow chew and when she's not actually eating. But rumination is the process when a cow swallows a lot of her feed very rapidly, but she doesn't actually chew it completely before she swallows it. And so this leads to her later regurgitating that feed to re-chew it, and that's what we call cud chewing. And this cud chewing helps further break down those plant and feed particles that she's eaten, and then re-swallow it for later breakdown by those microbes in the rumen. And so there is a little trick that I learned in undergrad. It's called the 3 rule, and cows will sleep a third of the time, eat a third of the time, and then chew their cud a third of the time. So 3, 3, 3. And then looking at why this rumen is so important for this animal, it stores large quantities of masticated feed material. And it's also home to billions of microbes that are absolutely essential for the breakdown of this feed in the rumen. So here are some images that I have of rumen microorganisms. This is a protozoa and a fungi, as well as this is a feed particle that you can see different microbes attached to it, trying to break down this feed particle, as well as other microbes, bacteria and fungi in the rumen. Just for some visual images. Okay. Any questions so far, Lauren? No, we're good. Okay. So just to look a little bit at the rumen ecology. So I have this visual picture kind of to look. So here's the esophagus where the feed is going to come in to here. And there's 3 different phases: the gas phase, the liquid phase, and the solid phase. So what's important about the rumen is that it's anaerobic, which means it's without oxygen. There's no oxygen here apart from very trace amounts that come in on the feed particles. And so looking at this, these feed, the solid phase that is present is the feed that accumulates within the rumen. And it'll be further broken down by these rumen microbes into the liquid phase before it gets passed onto the omasum and into the small intestine. And then the gas phase. So this fermentation process that occurs with these rumen microbes actually release these gases into this upper portion of the rumen. And I will get further into what happens here, but these are fermentation byproducts that occur when this feed is fermented and broken down. So another thing to note is that the rumen is essentially falls upon the pH environment of it. So the pH typically ranges from 6 to 7, and this is very important for microbial establishment within the rumen and to basically keep it at an optimized environment. So too low of a pH can impact and harm the bacteria and microbes that are in the rumen, and too high of a pH can do the same. So it's very important to have a pH that is in optimal range for these microbes to ferment and to perform at the best of their capability. And then as far as temperature goes, this rumen environment is held at 39 degrees Celsius or 102 degrees Fahrenheit, so fairly warm. And in these 3 parameters here are very important to making sure, like I said, that this rumen is in optimal condition in order for these to be fermented, the feed to be fermented and utilized by all of the microbial species present. So just a touch between the symbiotic relationship between the cow and her microbes. And I kind of wanted to reach out to you guys and see what you envision symbiosis, or what a symbiotic relationship is.
So you guys can type in the chat what you think that might be. And I know you all have some ideas. Benefits to both parties. Let's see. It's when two things help each other. Anyone else typing away? Yep, two organisms benefit working together to improve their chances of survival. Symbiosis is the act of two or more organisms being dependent on one another. Both organisms benefit. And again, when both parties benefit, I think they get this.
Yes, that's very true. Great job, everybody. That's correct. So yes, a symbiotic relationship is when two separate organisms both have a mutualistic benefit for each other. So great job for all of you guys who answered that correctly. And so we're kind of going to go into how the cow benefits her microbes and how the microbes benefit the cow. So this is just a brief synopsis, but basically we have this saying that when you feed a cow, you're not actually feeding the cow, you're feeding the microbes in the cow. And in turn, those microbes will feed the cow. So essentially this feed is broken down and you're feeding these microorganisms, and then in return those microorganisms provide energy and protein for the animal. As part of this relationship. So my question to you guys, and I have another one for you. How does the cow benefit her microbes?
So there's always just the delay while they're typing. So think
No,
about this, type away. How do you think the cow benefits her microbes? Ezra says the cow gives them food and a place to live.
Okay.
Any other thoughts? Let's see. We'll give it a sec. Yep. She provides food and a warm space for them. The cow both provides the food and the environment. I see a theme coming here. Jackson says by providing the microbes with nutrients.
Yeah, that's great. Very good. Okay, yeah, that's awesome. Yes, so that's definitely a portion of what she does for the cows, for the microbes. So she ingests feed to stimulate microbial fermentation. So a lot of what you said. So she's able to bring those nutrients to those microbes that are living inside of her. And she also ruminates, like I talked about, her cud chewing, to grind the feed to a smaller size. And this assists in microbial fermentation. So essentially, if those plant particles are broken down more, then the nutrients inside those plant particles are more accessible to these microbes that are trying to ferment those products. And so it's very important that she chews and breaks down these feed particles to facilitate fermentation. And then she removes microbial fermentation products from the rumen, which ultimately could impact the growth and replication of these microbes. And so we'll get into that a little bit, but essentially the fermentation products will be, sorry, there was a chat that popped up. These fermentation products will be produced once these microbes utilize and break down these substrates that they're getting from the feed that the cow provides to them. And so just like we as humans have waste products from eating, so do these microbes. And so have a few questions that I can answer really quick, Lauren.
Yeah, so Lori asks, what are the advantages of being a ruminant animal versus a monogastric animal?
So monogastric animals are typically omnivorous, which means that they can consume both plant and meat. So being a ruminant animal, this fermentation process that occurs, that happens, is really important for animals that consume large quantities of forages. And we'll get into why that is a little bit when we look at what the structure of what a forage is and why it's difficult to break down. So that is the benefit of being a ruminant animal, is that they have this pre-gastric chamber in which they're able to ferment these forages to break them down, which monogastric animals do not have. So that's the difference between the two.
The next question is the relationship between the cow and her microbes endosymbiotic since the microbes are inside her?
Potentially. I have never heard it been called that. I just look at it as a symbiotic relationship. It's what I've been taught. But I definitely can look into that.
Okay. And here's one that's been showing up in the chat. So inquiring minds want to know, do ruminant animals explode, or do their stomachs explode when they die? Is that fact or myth?
They do not explode. There is accumulation of gas buildup sometimes when there is nutritional deficits and things that occur in the rumen. There can be gas that explodes, and that's called bloat, where they'll have too much gas accumulation that can escape from the rumen, but it doesn't per se explode. And when they die, it's those microbes and everything in them will die with them. It won't, there's no explosion occurring.
And then the
great
last
question, great question.
Yeah, like I said, inquiring minds want to know.
Yeah,
The
no, that's a good
question we have at the moment, Jackson wants to know, how does a cow get her microbes?
That is a very good question. That is a very good question. So when a calf is born, she's born with some microbes that are already present in her. They're just already there. And then through her experience through life and her what she accumulates, whether that be because she was licked by her mother or she ate something with other microbes, these microbes slowly build, and there is a specific population that is established between all cows. And so whatever she's exposed to as she grows older will further facilitate the growth of these microbes in her rumen. If that answers your question. So it's all about exposure. Okay, we'll answer more questions coming up. We'll do this. Okay. So my question again to you guys is, how do the microbes benefit their cow? Since we...
All right, so the first question you already answered, how did the cow benefit microbes? Now it's reverse. How do the microbes benefit their cow? Eva says they digest her food.
Okay.
Give it a sec, see who else is typing away. What else do you guys think? Ezra says they ferment the food to help with digestion.
Good.
Shterna says they aid digestion, and Lori says digest food. We got a theme going again.
Okay. Okay, awesome. Yes, so that is definitely a part of it. They help digest the food. That's a big part of it. So like I said before, that symbiotic relationship is they're responsible for feeding the cow. So before I talked about these byproducts that are produced when they're fermented, and these are called volatile fatty acids. So when carbohydrate breakdown occurs from plant material, the rumen microbes take in those carbohydrates and they ferment them into what's called volatile fatty acids, VFAs. And these volatile fatty acids are very, very important because these are essentially what are going to provide the most nutrient source to the cow. So they're the primary energy source for the cow. And these volatile fatty acids, acetate, butyrate, and propionate, will be produced by these microbes. And they're also important for the primary, as the primary fat source in milk, so that those help get converted into milk and the fat in milk later on. And then additionally, these microbes in the rumen are able to utilize non-protein nitrogen. So for us as humans, we are able to derive our nitrogen sources from protein because nitrogen is a component within protein. But these microbes are very special in that they can use nitrogen that's not derived from protein and basically make their own microbial protein inside their cells. And so what happens is these microbes don't stay forever in the rumen. They keep repopulating, but a lot of them will die off. And those that die off will get passed down into the small intestine and the abomasum. And the protein within those cells that they created will actually serve as the majority of the protein source that the cow will get. So approximately 75% of the protein that she gets in her diet is actually from these digested microbes that she had in her rumen. Moving on. So now I just kind of want to touch base on what these microbes do in the rumen and what we can make them do to perform better. So looking at the microbial population as a whole, there's billions of microbes in one cow's rumen, and those include bacteria, protozoa, fungi, and different types of viruses. And then all of these together form a complex of different communities, and they all work together to aid in the feed degradation that's consumed. And so looking at this chart, you can see that, and they did this as a comparison of how many humans on Earth there are. But bacteria, you can see the amount of bacteria in a single rumen is more than the amount of humans that we have on Earth. And then we have protozoa, about half that number, fungi, and then mycoplasma, which I won't get into, and viruses. So many microbes in the rumen that all contribute to this feed degradation. So here are some photos that are, they're electron micrographs that were taken of different feed particles and these microbes breaking down each of these feed particles. And you can see in size for comparison, micrometer is very, very, very small. So these are very enhanced images. So as far as rumen bacteria is concerned, there's about 10 to the 7th to 10 to the 12th mil per milliliter of rumen fluid. So if you think of a milliliter, it's a very, very small amount. And there's billions just within that milliliter of rumen fluid. And so of the bacteria, there are different groups of bacteria that perform that have different functions that they provide in the rumen, such as cellulytic, amylolytic, proteolytic, lipolytic, and methanogens. So much like our society has a culture of working together of different jobs to perform, such as doctors, lawyers, they all have different functions to provide within our society. And this is the same. All of these bacteria as a group and a whole have different functions that they provide within the rumen to help break down these different nutrient components that are there. And they also, bacteria, they're the most abundant, but so therefore they provide the largest proportion of all the microbes of feed degradation. And so looking at these different groups, and these are just a few, they're not in total of what they are, what all of the bacteria are in the rumen. But lytic means breaking down. And so if we look at this, is there any way that you can kind of determine what the action of this each of these groups of microbes are based on their name?
So if lytic means breaking down, what do you think I can't say it, cellulolytic means? What does
Or
cellulo mean? Or what does amylo mean? Or can't
proteo or
proteo,
lipo.
lipo. You guys have a guess? Alice says breaking down cells.
Okay.
Anyone else have a thought? What are they breaking down? I feel like I want to play the Jeopardy theme song right now.
That's okay. That's okay.
Oh,
if you
there we
don't
go. We have breaking down methane and breaking down cells, breaking down proteins.
Good. Okay. Very good. Okay. Yeah. Yeah, very good. Methanogens actually are creators of methane, so they don't actually break it down. Lipolytic is fat. Lipo means fat. Proteolytic is breaking down of proteins. And amylolytic, the breaking down of starch, and cellulytic is cellulose, which is a component that makes up fiber in forage particles. So good job. Those are great guesses for sure. So here's just a chart of what each of these components are and the type of bacteria they have. So cellulose degrading bacteria, you can see, and these are just a few. These don't encompass all of the bacteria that are actually there. Actually, a lot of the bacteria that are in the rumen still hasn't even been discovered yet because there's so many, we don't even know all the functions that's there. But these are just a few that have been identified, such as Fibrobacter succinogens, and this is a bacteria that degrades the cellulose and the fiber that's in the rumen. And then you have those different byproducts that I was talking about that they produce in order to help provide those energy sources to the cow. And then the same for amylolytic bacteria, which is starch, fat breakdown, and then several others, including methanogens, which help produce methane, and I'll get into that as well. So the other microbial group I kind of wanted to touch on with you guys that are really important are Rumen Protozoa. And so there are far less Rumen Protozoa than there are bacteria. So about 10 to the 5, 10 to the 6 per mil of rumen fluid. But they are the largest rumen microbe. So these guys can range anywhere between 20 and 200 micrometers. And rumen bacteria, for example, could be 5. So they are much bigger if you're comparing, say, a shark and a small fish. That would be the equivalent of this. And so these guys actually will ingest bacteria to feed themselves, but they will also help ferment the feed particles that are in the rumen. And these guys utilize soluble sugars and polysaccharides that are broken down from the feed particles that are consumed. And then they will engulf whole starch particles. And this helps in the decline in pH by basically storing that in their cells so that the bacteria can't ferment that starch too rapidly in the rumen, and basically bring the pH of the rumen down. And then another key important microbe in the rumen is rumen fungi. And essentially, they are in smaller proportions as well, 10 to the 6th to 10 to the 7th. But these guys are very, very important because they contain very active enzymes that can help break down cellulose and hemicellulose. So they have very high efficiency when it comes to breaking down these fiber particles. And they help, this in turn helps improve the rumen fermentation by increasing that digestibility that's there, which in turn can help increase the production and performance of the animal whenever the energy and protein goes up from the increased digestibility. So my question to you guys is, what is the purpose of an enzyme?
Take your guess. What do you think? Give a moment as the typing and the delay. And that's okay, Eva. I can't remember what that is, she says.
No, that's okay.
It's totally okay because you're here to learn.
Yeah, exactly.
Egan says breaks down starches, corns, or something, and Jackson says to turn starch into sugar.
Okay. Those are really, really good guesses. So actually enzymes, from a chemical standpoint, is a substance that acts as a catalyst in living organisms to accelerate the rate of a chemical reaction. So these enzymes—
I gotta say, I just want to give Lori props. You don't see the chat, but Lori said that they speed up chemical reactions in your body. So good job there.
Yeah, great job. Yeah, I didn't see that one. It must have came in late, but yes, great job, Lori. That's exactly what it is. So essentially it is a substance that can help facilitate these reactions to happen at a more at a faster rate. And so this is what happens in the rumen. These microbes have these enzymes that they produce in order to make the breakdown occur at a faster rate. And these bacteria are very special in the sense that they, we as humans don't contain those enzymes. So those bacteria that break down the forages are very important because they're required in order to break down some of the components that we ourselves don't utilize. And to further that, so if we're looking at a plant, any kind of plant or forage particles, you have the plant cell wall, and this cell wall is very rigid and contains all of these microfibrils that basically hold the structure of the plant. And these contain cellulose, lignin, and hemicellulose, and they basically all branch together in order to form that rigidity in the plant of these cell walls. And so these cell walls are very difficult for humans to break down because we don't have those necessary enzymes to break down things such as cellulose. And what the cellulose is essentially this long chain of polysaccharides, which just means multiple, poly meaning multiple, and saccharides meaning sugar. So this is a long chain of glucose all linked together, and their linkages that they have between each glucose molecule is what makes them cellulose. And if I go to the next slide to show you guys. So this is the structure of a cellulose molecule, and this is just a bunch of glucoses that are together, and this is in a plant, the plant cell wall. And so essentially these bonds that are linking these different glucoses together are very difficult to break down on a normal basis. So these cellulases, which is an enzyme, are used to basically break these bonds to make them single glucose molecules so that those can be utilized and fermented, because they're not able to be in this state. And so these microbes, especially those of cellulitic bacteria, contain a lot of these cellulase enzymes that they're able to use on these plant particles to basically break these bonds down in between each of these glucose molecules, so they're able to utilize those substrates for an energy source in their cell. So if we're looking at basically the proportions of food that we provide, so the amount and the groups of bacteria that are in the rumen are dictated based on diet. So the more forages or cellulose that you provide, the more cellulitic bacteria that are going to be present there, because you're providing those specific substrates to that niche of bacteria. So if we're looking at this, 80% hay, 10% soybean meal, and 10% barley, this hay, which is has so much cellulose and cell wall portions within the hay, is going to facilitate the growth of these cellulose and cellulitic microorganisms to produce these are volatile fatty acids. Those are the byproducts that I was telling you about for the energy source. And then you also have your proteolytic and lipolytic bacteria that help break down those portions of the feed. And then you also have your starch and sugar degraders, your amylolytic bacteria, and those are going to produce VFAs as well. The difference is that the starch is far less complex to break down than the cellulose. It's much easier for these bacteria to break them down. So they're able to produce these VFAs much more quickly than the cellulose degrading bacteria. So they're in a much slower rate because it's so complex to break these bonds of the cellulose, whereas the starch is much more simplistic. So if we were to decrease the amount of hay and increase the amount of barley, then there would be increased amount of starch and sugar, and that we would get more VFAs at a faster rate. And so when we look at this pH scale, it's important to note that balance is key in formulating diets of ruminant animals, because if you provide too much of that starch, those VFAs are going to build up, and they're volatile fatty acids, which means they're acids, and they'll bring down the pH of the rumen. And so if we let that accumulate too quickly, by feeding all of this starch, then it's going to make the rumen very acidic, which can have negative impacts on rumen health and the bacteria that are there. So like I said previously, that if the pH gets too low, a lot of these microbes and bacterial groups in the rumen can't function properly at that low of a pH. And so it's very important that you have both, in the sense that you get the amount of VFAs that you need to maintain production and performance in the animal, but you're also facilitating how much is released by incorporating the amount of cellulitic organisms and having kind of a slow release, so that you're within that pH realm in order to facilitate optimized microbial degradation of the feed particles. Hopefully. From here, does anybody have any questions? It's kind of a good stopping point.
Yeah, there are a couple of questions. Sterna wanted to know, can you have a microbe deficiency?
Yeah, so that's actually a really, really good question. So yes, there are cases where you can have a deficiency of certain microbial groups. So like I said, if you're feeding large amounts of the starch and these pHs go down, and the pH goes down, that's going to negatively impact the cellulitic bacteria, which means all that fiber that you're trying to degrade is going to go down because that group of microbes have been decreased based on the pH that's in the rumen. And so you can have deficiencies of microbes where the function of the rumen does not perform properly, either because of poor diet and the balance that you have between those aspects, or there's some like antibiotics and different type of medications or along those lines that can make a deficit in a certain microbial population in the rumen. Hopefully that answered your question.
Yeah, and then the next question, it just says, aren't viruses harmful? And I think that goes back to that slide where you were, I'm going to guess, where you were showing the bacteria and the other stuff in the humans and their viruses.
Yeah. So viruses have different functions, and yes, a lot of viruses that we think of in our mind are harmful viruses to our health, but they're actually viruses that have positive impacts as well, just like bacteria. There are harmful bacteria, and there are pathogenic bacteria, and there are helpful bacteria. So it's the same for viruses. Not all of them are bad. We just tend to hear more about the bad ones than we do the good ones.
And the next question, who is in charge of naming the various bacteria in the rumen? Since there are many that haven't been discovered, if you found one, who would be able to name, would you be able to name it yourself?
That is an amazing question. So as far as I know, the majority of these microbes that are discovered are basically named, a lot are named after their function or the group in which they fall in. And so I don't necessarily want to say that, yes, you can name it after yourself, because I haven't really seen anybody name the rumen microbe after themselves. But that's a great question. I think that more often than not, they name it based on the functionality of what that microbe does.
Okay, good.
Okay, awesome. Great questions. I love those questions. Okay. So now talking about what we can do to improve the microbial performance in the rumen. So like I said, diet is crucial in balancing those different groups and communities of microbes in the rumen. And so that is the true key to doing that. And once you've done that, that opens up this realm of adding different feed additives on top of their normal diet to either enhance or target specific microbial groups in the rumen. So that includes things like phytochemicals, probiotics, ionophores, fibrolytic enzymes, and yeast. So just to kind of see, do any of you guys know what these products are, or what these mean? Do you know what a phytochemical is?
Or maybe any of the other ones.
Yeah, or any of the other ones, just kind of.
What do you think probiotic might mean? Break down the word probiotic. Or Lauren had said lytic means, oh my God, now I'm gonna space. We already learned what lytic means. And
Yes.
then fibro. So Eva says a probiotic is a megavitamin, right? Simon says yeast issued in bread.
Yeah.
Ezra says supporting biotics or probiotics.
Yeah, exactly.
And that's okay. And Bowden says I'm unsure.
No, that's okay. That's okay. Good try, everybody. So just to touch, yes, probiotics are basically healthy bacteria that you can use to stimulate. So we as humans take those to help stimulate our gut microbiota. And so that's also true for rumen microorganisms, as you can facilitate them by providing these live microorganisms to help basically stimulate the population that's already there. Fibrolytic enzymes, so breaking down fibers. So those are just providing additional enzymes, fiber degrading enzymes in the rumen to help kind of facilitate that fiber breakdown. Ionophores are not an antibiotic, but they function similarly to an antibiotic in the sense that they target specific parts of a cell. So the bacteria have different cell structures, so you can target that specific cell structure in order to either limit or promote a specific bacteria based on their cell structure. And then phytochemicals are very cool, and there's a lot of research going into phytochemicals, but basically those are the chemicals that are naturally found on plants. So each plant has a different chemical that they might produce, such as tannins, saponins, different chemicals, and we're researching how these different chemicals, if you feed these plants to this animal, how they impact that rumen microbiota and change the aspect in which the fermentation products are fermented, or the different bacteria that are actually there. And then yeast, like whoever said bread, very good. It's very similar to that. So it's the same type of yeast that they use in like making bread and stuff like that. But you can utilize that to actually stimulate the rumen microbiota that's in the rumen as well. So there's a lot of research going into if you feed yeast to a cow, how can that impact those microbes in there and basically the products that they produce at the end. So great job, everybody. You guys had a good impact with that. So one target that is very big in cattle nutrition is methane production, or targeting methanogens. And so to utilize as an example for like different functions that we want to use, use these feed additives for. So methanogens are a type of bacteria that live in the rumen, and they basically utilize the carbon dioxide and the hydrogen that's produced in the rumen during this fermentation product. And you can see up here in the top of the bubble. But basically they take those two gases and they bind them together to make methane. And that helps these gases escape from the rumen and keeps them from building up in the rumen. So not only is this harmful to our environment, it's one of our greenhouse gas emissions that we're trying to limit by utilizing these feeding strategies. So not only is it harmful to our environment, but it also causes an energy loss in our cattle. And we want to utilize our feed at its highest potential to turn into energy so that there's no waste from that energy that we're providing. And so approximately 5 to 10% of the energy that the cow would get is lost through methane. And so trying to come up with these nutritional strategies and these feed additives to kind of use and target this specific group of microbes in order to minimize the amount of methane that's produced in the rumen. And so hydrogen, these hydrogens are super acidic. If these accumulate too much in the rumen, they can bring the rumen environment down. So there's also this symbiotic relationship between methanogens and the other microbes in the rumen, because they help maintain that pH by removing these hydrogens from the rumen environment. So these methanogens have this positive relationship on these other microbes. And so it's important to kind of touch base that this is still an important mechanism that happens in the rumen, even though it's not environmentally or energetically favorable mechanism for us. And and then, like I said, it has a symbiotic relationship with other microbes, primarily protozoa. So a lot of those really big microbes that I was talking about, these methanogens will sit on the cell surface of this protozoa. And essentially, when the protozoa ferments its feed and releases the hydrogen as a byproduct, those methanogens will come in and basically take it away, and making the environment more favorable for that, for those microbes that are there. Okay, so now I just kind of want to touch base a little bit about what my research is. So my research encompasses continuous culture, and basically that is an in vitro model. So basically it's not in the animal. So we're actually able to take some of the rumen contents from the cow, and that has all the microbes in there as a subsample, and basically test these different feed products in these fermenters in order to measure their different outcomes that we would somewhat be able to measure in the real live animal. So what my research looks at is yeast supplementation. And this research is basically looking at a few different components, such as increasing cellulitic bacteria. So yeast have been shown to help improve cellulitic bacteria and function in the rumen, which ultimately can help in increasing the fiber digestibility, and also the production and increasing production and performance, because the more digestible the feed, the greater end products that we receive at the end. So yeast supplementation has a few mechanisms in which they've been shown to increase cellulitic bacteria. And then they've also been shown to increase lactic acid utilizing bacteria. So bacteria in the rumen, there are a few different groups that produce lactic acid, and lactic acid is a very, very strong acid that can bring the pH down in the rumen. But in order to circumvent this, there's actually lactic acid utilizing bacteria. So bacteria that come in and utilize the lactic acid that is produced, and convert those to VFAs, which is a weaker acid, and able to convert that into further end products. So this helps modulate the rumen pH in those VFAs that are produced if we're able to target these lactic acid utilizing bacteria and basically stimulate them to work a little bit harder to uptake this lactic acid in the rumen. And then there's also been some studies to suggest that that yeast can reduce methane emissions. So there's further research into looking into the mechanisms behind that. And then, so this is just kind of the process that we do. So this is a cannulated animal. We have a few at the dairy farm at the university, but essentially this cannula allows access to the rumen. And so we're able to take those contents out and to put them in the jar to measure these different bacterial simulations in vitro, which helps us test these different products before we ever have to put them in the live animal, because we don't know what they're going to do yet. So that helps us kind of monitor what these feed products can do. So here's just a few pictures of that, the removal of rumen fluid with all the bacteria in it, and then our lab group doing collections out at the farm. And just some measurements that we're able to take that are in the lab that are similar to things that we would look at in the cow. We're able to measure the pH of the environment and the temperature. So that's regulated. You can see these little red jackets on here are temperature regulating heat jackets. We're able to measure the VFAs that are produced that get collected in this blue container, as well as the microbial protein and ammonia that's produced. We're able to see how much of the feed that we put in here is degraded, so what the digestibility is. We're able to look at the microbial profile and see how that changes based on supplementation. We're able to measure how much protozoa is in the rumen, and also utilizing a methane analyzer in order to monitor how much methane is produced in each of these systems. This is just a picture of me with the continuous culture fermenters, just doing a little bit of lab work. And then just looking at our overall outcomes and goals that we have from this research project. So we want to basically establish what the microbial response and what their functions are from supplementing with these yeast products. And then eventually we're hoping that that can further down the road lead to in vivo studies and actual animals to determine how what production parameters such as milk and the impacts that they can have in the actual animal. And then overall, just looking at how this can improve animal health, their performance and their production, as well as sustainability measures such as methane emission reduction. And with that, I ask you if you have any questions.
That's great. There are some questions in the Q&A, but before we get to those, I'm just going to launch our feedback poll. There's 2 questions: how you thought today's cafe was, and then more importantly, did you learn anything? So scale of 1 to 5, 5 being excellent. So just take a couple of seconds here to give us the feedback, and then we'll get to the questions. And you can add in some questions if something has come up that you would like to ask Lauren. We'll just give it a little bit, let everyone answer. And then we have about 3 or 4 minutes for questions.
Okay, perfect.
So if for some reason you can't see the poll, please put your rating in the chat, but we do like to get as many responses as we can get. It's very helpful to us. Going to give it about 15 more seconds. Hopefully, if you haven't responded yet, you'll go ahead and do so. Okay, I'm going to close the poll and let's go to the questions. So Amber wants to know what happens if the cow has too many microbes?
There's not going to ever be too many microbes. There might be an imbalance of one microbial group to the other. But like I said before, there's a way for these microbes eventually will die off. There's a life cycle that occurs, and those microbes that die off will be ingested by the animal and its nutrients utilized by that animal further on. So there's never going to be an overabundance of these microbes in the rumen. There's a natural life cycle that continues.
Great. Sharna wants to know, can you give an example of how people are trying to lower methane release while keeping the cows healthy?
Yeah. Utilizing those phytochemicals from different plants can help target those methanogen groups. So a lot of studies right now are looking at feeding seaweed. So there's some chemical components within seaweed that have shown to inhibit a lot of those methanogens, and it's not harmful to a lot of the rumen environment as a whole, but it just specifically targets those enzymes within the methane producing bacteria. And so we're utilizing different of these nutritional strategies in order to target those and basically proliferate certain bacteria among the maybe non-beneficial bacteria, such as the methanogenic bacteria.
Does the rumen affect milk? For instance, how it tastes and what nutrients are in it?
Absolutely. Yes, that is an amazing question. Yeah. Also, beef cows, milk, and meat as well. It definitely does. So those end products that are produced, the volatile fatty acids, those greatly impact the amount of fat and protein and different nutrients that are in the milk that you're going to get. And so a lot of research that goes into this is trying to formulate and optimize the diet to where we can feed less feed to the animal, more high quality feed, and get a greater product at the end, such as milk and meat products. And so these end products and these certain bacterial groups that we're able to target most definitely influence the end product that we get at the end. So that was a great question. Great job.
Great. And I'm going to ask at this point, no more questions come in because we have 4 and we're already over time. So we're just going to answer the 4 questions that are here. Will speeding digestion speed insulin production?
That's a great question. So insulin is released in the body in response to glucose concentrations that you have. And so a lot of these volatile fatty acids are transported in the blood, are not identified as glucose as glucose. They're a different form of in the rumen. But yes, anytime you increase the feed consumption, you're always going to see a positive correlation in the increase of insulin that's released, and that's across all animals. But there's slightly more complex mechanisms that go into this. So I won't be able to dive any further on that, but that is an excellent question.
As Sterna is saying, she's still not fully understanding. So what happens to gases if they aren't being released? If you can.
Yeah, no, that's okay. So bloat is when all these gases accumulate in the rumen and can't get out, and it can be very harmful. And you see it a lot in calves that aren't fully matured yet, but it can essentially kill the animal. And so it's very important. And there's different devices that veterinarians use called trocars that can kind of release the gas accumulation. But a lot of it is trying to manage the nutritional aspect of it so that you don't get that accumulation of buildup of those gases in the rumen. But yes, they can be harmful. And if too many gases accumulate, can kill the animal.
So this is going to be a simple yes, no. I know the answer. Does having, in your research study, the hole that you have in your animal, does it hurt? Does that entrance to their stomach hurt?
Hurt the animal?
Yeah.
No. Yeah. No, it does not. It's a very it's a natural. They don't feel it. In other words.
Yeah. Nice. All right. In 10 seconds or less, do you have any cute cow stories?
Yes, being on the farm and being chased by a herd of little tiny calves all around the calf barn because they're so interested in you. They just love to follow you around and lick you and smell you. So yes, there are many cute cow stories.
Nice. Well, everyone, let's thank Lauren for her time today and all of her knowledge that she shared with us. I mean, you know, cows are so cute, and then look at all we can learn about them. So thank you all for joining us today. Hopefully we'll see you next week at next week's Science Cafe. But until then, everyone have a good night.